folks, this is Dr. Powell. Welcome to Anatomy and Physiology on Demand. This is a lecture 11B part 2, where we are going to learn about the electrophysiology of the neuron, which is how neurons communicate with each other. And so to understand that, there's a couple of terms we need to understand: electrical potentials and how that differs from a current. So an electrical potential is just that. It is a potential, a difference in charges between point A and point B. And so we have this potential energy, we have this potential change that can happen between these two. And when that change happens, that is what a current is. In other words, we will have a flow of charged particles from point A to point B, and we measure this in volts, or in neurons, we measure it in millivolts, or in thousands of volts. Okay. So in order to measure it in the cell, we have our reference electrode outside the plasma membrane, and that is set to a volt level of 0. And then we put a little micro electrode through the phospholipid bilayer into the cytosol, and the tip is measuring the difference there. And in a resting axon, that would be negative. So to review, normally we have sodium outside the axon, potassium inside the axon, and that difference is kept with the sodium-potassium pump. And so when the action potential hits, which is in the second column from the left, the sodium gates open, sodium enters the cell, potassium gate, and that depolarizes it. Potassium exits the cell when the potassium gates open, and that is going to cause repolarization. And then this change, this negative polarity, will be maintained with the sodium-potassium pump. So go back and look at your old slides if you need more detail on that. So whereas we have talked about action potentials and voltage changes in the different types of muscles, so far I've told you don't bother learning about the numbers because they differ between skeletal, cardiac, and smooth muscle, as well as neuron. So for the neuron, we are going to be learning some numbers. Okay. So the first number we're going to learn about is the resting membrane potential. In other words, what is, when the plasma membrane of the axon is at rest, what is the measurement? Okay, so if we set that extracellular fluid at 0 and we put that microelectrode into the cytosol of the axon, it will measure around -70 millivolts. So this is our starting point, also known as the resting membrane potential. So what this means, since it is negative, it means there are more negatively charged particles inside the membrane than outside the membrane. So yes, we've got chloride on both sides, but we have a lot more anions inside than we do outside. So why is it -70? There's 3 factors: ion diffusions, down concentration gradients, some selective permeability, and then some attraction. So I want to talk about specifically sodium and potassium and how they affect this resting membrane potential. Potassium has the biggest influence because the plasma membrane is most permeable to potassium. Not only can it get through when those channels are widely open, but as we talked about before, potassium channels tend to be leaky. And so potassium can continue to leak out down its concentration gradient, and especially if there's more negativity in one place, it will leak because it will be attracted. And so eventually we will have some type of equilibrium, and at that point there'll be 40 times more potassium inside the cell than there will be outside the cell. And if this was the only thing that determined the resting membrane potential, instead of being at -70, it would be at -90. So that brings us to the influence of sodium. As opposed to 40 times more potassium being inside the cell, there's about 10 to 15% more sodium outside the cell. Because it's attracted by negative charges inside the cell, it will trickle into the cell, reducing the voltage. And so that would bring the voltage back up someplace closer to a -75 if those were the only 2 influences. And so the last influence is that sodium-potassium pump, where it's pumping sodium in exchange for potassium, and that accounts for about the last 3 millivoltage difference in the two. And this pump actually uses about 70% of the energy of the neuron, and it's that constant need for glucose / lactate and oxygen. Okay. So the next concept is something called a local potential, which we have not talked about before. And what this is a very short range, a nearby, a neighborly change in voltage. So when the sodium rushes into the axon, it actually will diffuse down its concentration gradient for a short distance on the inside of the plasma membrane, causing this little wave of excitation, this tiny little current spreading out from the point of stimulation. So the drawings there show it spreading out to the right and left, but I want you to realize it's not just in one plane. It's actually spreading out like when you put a little pebble in the water and you see the ripples going out in 3D. But what is important to realize, this is a very short range change in voltage. Okay? And so it dies out very quickly, which is what this picture is showing. If you have these microelectrodes at 3 different places along the axon, once you have the stimulus, you can see the action potential generated at that point, but then you can see further on, everything is sub-threshold, so you're not going to be having sodium entering the cell. So looking at local potentials, it has some very unique characteristics, some of which we've mentioned before and some of which we haven't. The first thing it is graded. In other words, it can vary in voltage depending on the strength of the stimulus. Okay? So the more intense, the stronger it is, the longer that ion channel is going to be open and the more that ion can go through. So this is not an all or nothing phenomenon. The second thing is decremental. The further you are from that point of origin, the weaker it is going to be. And so that is what that upside down mountain or the drawing underneath it is trying to represent. So take a moment to look at those pictures as well. The third point is it's reversible. As soon as that stimulation stops, the local potential ends. The cation is going to diffuse out of the cell very quickly. We're going to be back to our -70 resting membrane potential. And the fourth characteristic is that this can be either excitatory or inhibitory. In other words, what we have always been talking about so far is what we see with acetylcholine, where it brings us from -70 up to the threshold at -55 and leads to depolarization. Okay, so we have raised the resting membrane potential. However, other neurotransmitters will even lower the resting membrane potential further or hyperpolarize the cell, which would mean you would have to have an even bigger stimulus in order to reach threshold. So that brings us to the threshold potential. In other words, what's the minimum voltage needed to open those voltage gauge channel? And in the neuron, we got to figure out how to get from -70 to -55 millivolts. And so if stimuli are not strong enough, they could partially bring us that way, but if we haven't reached -55, you're not going to get your action potential. If it is reached, we will generate the action potential as seen in the top picture. If it does not, then those stimuli, as seen in the bottom picture, are called sub-threshold stimuli. Continuing on, let's assume we have made an action potential. In other words, we have depolarized past -55. Well, the first thing to realize is this cannot happen everywhere. It only will occur where we have a very high density of these voltage gated channels so that we can have massive amounts of ions to be moving. And in fact, the density is about 5 to 10 times higher than it is at other parts along the axon. When the action potential occurs, we're going to have a rapid upward or else positive change in voltage followed by an even more rapid downward change in voltage. So at threshold, those sodium channels are opening really quickly and massive amounts of sodium are rushing into the cell. I want to point out that at the same time, the potassium channels are actually opening, but they are very slow to open. So potassium enters the cell slower. So it's not exactly like we talked about before when I did the little pantomime and we had sodium gates open, sodium rushes in, sodium gates closed, then potassium gates open, potassium rushes out, that both gates were actually opening almost simultaneously. We just had a lot of sodium running in initially. So when the sodium enters the cell, that's going to even further depolarize the membrane. Once it gets to a 0, that inactivates the sodium gates and they're going to start slamming shut. Now, it's not instantaneous, which means it's going to overshoot and peak somewhere around 30 or 35 millivolts in the positive range before the polarity gets reversed. And the big influence will be potassium exiting the cell, causing the repolarization. So this continues quite a bit longer. So you can see the potassium gates are open much longer than sodium gates. And the more potassium exits, the more negative it's going to get. And by the time they close, remember they're going to be leaky even when closed, we are going to overshoot the minus 70 millivoltage of the resting membrane potential. And we are going to be hyperpolarized. So looking at the characteristics of an action potential and comparing them to those 4 or 5 characteristics of the local potential I mentioned before, the action potential follows the all or none law. If the stimulus depolarizes the neuron up to threshold to that minus 55, that neuron is going to fire at maximum voltage. Secondly, it is not graded. If we are above threshold, a larger stimulus does not produce a larger voltage, as seen in this drawing. Thirdly, it's not decremental. In other words, it's non-decremental. What does that mean? As we continue down the axon, the last action potential that is generated near the axon terminals is going to be just as strong as the first one that was generated by the axon hillock. I want you to check on your handout because on this one where I have the word axon hillock appearing on my slide, I think I have axon terminal written on yours, so that was a mistake. So make sure you check it and correct it if you are looking at one of the older slides that said terminal. And so take a moment to look at the picture at the bottom right, because that is basically showing action potentials at 3 different areas along the path of the axon, so you can see what happens with it. And the last characteristic is the action potential is irreversible. Once it stops, once it begins, it cannot be stopped. In other words, if we're going to reach threshold, the action potential goes to completion. You cannot stop it halfway up. So the next concept has to do with refractory periods, which we have talked about before. And I want to point out the refractory period in the neuron only refers to that one small area of the axon membrane where we have already started the action potential. We will have an absolute refractory period as well as a relative refractory period. So the absolute refractory period basically starts from the time you hit threshold at minus 55 and includes all the depolarization time, no matter how high you go, and all the way back down until you hit the resting membrane potential in repolarization at minus 70. So this has to do when sodium channels are inactivated. So the first part of this are already open, so you can't open them if they're already open. And then they're in the process of closing and they're just not able to be reopened again. Also realize that most of our potassium channels are open during a majority of this time. The relative refractory period is then due to continued outward diffusion of potassium is going to start at minus 70. We have now reached the resting membrane potential again, but remember we are going to continue having potassium leak out, so we are going to hyperpolarize, so that the relative period starts from the beginning of hyperpolarization and ends at hyperpolarization. So what does this mean? It means that an impulse during this time would have to be stronger in order to reach minus 55. In other words, to go from minus 70 to minus 55 would require an impulse to move you 15 millivolts. But to go from minus 90 to minus 55 would require a stimulus to move it minus 35 millivolts. So a much stronger stimulus. So if you had one that strong, yes, you could have another action potential. So let's look at this diagram. Start with the one on the left. This is the distorted one, which allows us to see all the details that we have just learned about. Starting on the far left, we start with the resting membrane potential of minus 70. Number one is showing us the effect of the local potential where we are reaching the threshold. And number two, we now have sodium gates opening. We're having massive sodium flow. We are depolarizing. We also have potassium gates open. Once we hit around number three, we have reached zero. Those sodium gates are starting to close. However, we still continue to depolarize to around plus 30, plus 35 millivolts. And then the effect of potassium loss takes over, and we are repolarizing all the way down in number 5. We go negative all the way to the threshold. We continue negative until we reach the resting membrane potential. And then we're going to start into the relative refractory period in number 6, where we are hyperpolarized. And eventually we will make our way back to minus 70, number 7, when we are at the resting membrane potential. Now, if we put that on the real time scale, that would give us the picture on the right, where you don't see any evidence of the local potential whatsoever. You just see this huge, almost straight up and down spike. Okay. So that's the first thing to note. The second thing to note is, see how long hyperpolarization is? That spike of that action potential is like 1 to 2 milliseconds, and that hyperpolarization is many milliseconds long. Whereas on the picture on the left, it looks like that local potential is longer than the hyperpolarization, and is actually longer than the actual action potential, depolarization, repolarization. Okay, so that's why the one on the left is so distorted, because time-wise, it's really not accurate. So the last thing is a distorted slide for you to go through and talk among yourselves or your friends, and to make sure you understand this material. And with that, that is the end of this section. And so understand this, and then I will see you for part 3 shortly.